Okay, can you shut it off now, please?
Sounds good, sure.
Yeah.
Start the recording. Here we go. So it's my pleasure to moderate today's iReceptor Plus seminar series. And as usual, we have two speakers. Our first speaker is Bryan Briney, who's an assistant professor at Scripps Research. This following a PhD at Vanderbilt. The thing to say about Bryan, he's just done an incredible amount of work in producing air-seq repertoires, both producing the data and analyzing the data. I know it mostly in terms of support of vaccine research, but there's other aspects to this. And today he's going to be talking about applying that to LASV infection. So please take it away, Bryan.
All right. Hi, everybody. It's good to be here. So I'm looking forward to this talk quite a bit because giving talks to things like the iReceptor community or the AIR community are so much fun because you guys are like my people and you guys are definitely interested in doing more of a deep dive into what we're actually doing and not just kind of like, we found this cool antibody, blah, blah, blah. You want to, so I wanted to talk a little bit about one of our projects that it's currently ongoing, which is to look at antibodies against Lassa virus. But I also wanted to talk about some of the new tools we're developing, particularly for a lot of single cell work that we're doing. So most of our work now is moving towards single cell technologies, as I'm sure many groups are doing as well. So just a brief overview. I don't think I have to explain much about 10x Genomics to you guys, but we've been working closely with 10x Genomics for the last year or two on a few new approaches, which some of them they're going to be commercializing soon, and then others that we are kind of doing on our own. So antigen barcoding is previously been published as LibreSeq by Evelyn Georgiev's lab, and Ian Setliff was the primary author on that. And so the general gist of the process is you somehow attach a DNA barcode to various protein antigen molecules, label B cells with those protein antigens, and then using the droplet microfluidics, whether it's 10x Genomics or your platform of choice, the individual antigens are dragged into those droplets along with the B cell if the B cell is able to bind. These then the individual barcodes can be amplified and sequenced, and then the corresponding heavy and light chain sequences plus the antigen barcode reads can be obtained. So there's kind of three simple cases shown here: one in which you have an antigen specific B cell, one in which you have a B cell that's not able to bind antigen, and one that is cross-reactive, potentially, so if these were different flu HAs, for example, you could identify not only just the flu-specific B cells, but also those that are broadly reactive across your panel of DNA antigens. So we've, what we think is an improved approach for this, primarily in terms of the sample prep and how we're making the DNA barcoded antigens. So I'm not going to go into a ton of detail about that. It's pretty straightforward. Essentially, we just do a site-specific biotinylation using an AVI tag, and then we can use, there's commercially available versions of this, but we make our own. We just have a streptavidin linked fluorophore together with a biotinylated antigen barcode. So then you have this very easy, almost like plug and play setup where you can have large panels of barcodes that you can then link to your antigens of choice, and then go from there. So one of the problems we're starting to see, though, of course, and I think everyone else this is not a huge insight that we've identified this problem, is that you're now developing these data sets that are different from anything we've seen before. So you have a huge amount of information that you can gather from each individual cell, some of it genetic, but what I think is even more exciting is we're moving into using genetic, super high throughput sequencing based readouts for what would typically be a functional assessment, which is, antigen specificity is a good example. So we can now generate a huge amount of data for each one of these cells. So linking those together is a problem, as is just the magnitude of the data we're working with. And it's in different formats than the tools that we've been using before are able to handle. So we've developed a package. So this is one of those things that if anyone here is working on computational stuff at all, you kind of start these weird naming schemes. So probably the most well-known is something like incantation. So you start with, you know, presto and changeo, and now you've locked yourself into magical spell names, right? So for some reason, all the tools that I've been working on have AB in there, like AB as an antibody. So we've made this one called SCAB, which stands for single cell analysis of B cells, which allows us to integrate all of these data sets and work with them in what I think are some pretty cool ways. So I want to talk a little bit about how this package actually works and how you can use it and why we think it's a pretty neat way to use to analyze this kind of data. So it builds really heavily on ScanPy and data, which are pretty closely linked. So if any of you have been working on single cell data analysis in Python, you're probably familiar with ScanPy. It's, I think, what will be considered the default. It's essentially the Seurat for Python. And what the ANData object is, it's a fairly complicated setup when you look at it this way, but it's actually super simple and easy to use in practice. So you have the core of it is what's called X here, which is a sparse matrix. This is essentially your gene counts, right? So on the vertical axis would be your cells, on the horizontal axis would be your genes, and you have then this matrix of gene counts for every cell. You also have what's this at the top called var and AgBCs, which are pandas data frames, technically. But what you can do with these is you can add annotations. Var is for each of the genes can have annotations, and AgBCs is each of the cells can have observations or annotations. So for example, here you can have different names for your gene. So if you have the gene name plus the common name, that's a good example. An example of the AgBC data here could be, for example, if you have multiplex data, you could have the donor assignments or the batch assignments here. So each cell can be assigned to a batch. And then there's also these AgBC P&M and var P&M, which are for multi-dimensional annotations of either AgBCs or var. So we're primarily concerned with this corner up here, the X, the var, and the AgBCs. So what we'd like to do is start appending data to this. It's not necessarily built into the standard single-cell RNA-seq approach, things like annotated antibody sequences, for example. So what we did is we used the pair and sequence objects that we already have worked on using our ABX toolkit. So essentially what these are is they're very easy ways to package together all of your annotated information for either a single sequence or a sequence pair, right? A heavy light pair or a TCR alpha beta pair. It's kind of agnostic in that way. And so we use those and we essentially just add that data to the AgBC data frame, which allows us to link each individual antibody pair or TCR pair to the cell from which it was derived. And it also links together all the gene expression information. If you have feature barcoding, if you have cell hashing, all of that stuff is now linked together at the cell level. So we've also included a variety of processing tools and visualization tools that we think are super useful for antibody specific cases as well. So to get a little bit into that, here's how we would start. So the API for SCAB is borrows pretty heavily from SCAB or from ScanPy, excuse me, which is I think is very well done. So I think as you'll see as we go through here, a lot of things that I've seen and liked, I essentially just blatantly copy, right? Which I think is the highest form of flattery in one sense, but also there's no need to reinvent the wheel if someone else has done a very good job of doing it. So SCAB has a read 10x matrix function. So we've essentially copied that same format, but instead of just passing the feature matrix, which is your gene counts matrix, we can also pass other things as well. So for example, we can pass the BCR path, which is your BCR sequence data. You can pass your BCR annotations, which in this case is the cell ranger annotations. And you can also pass a format, which can be so your BCR data can either already be annotated or it can be just the straight FASTA file that comes right out of Cell Ranger. So the idea of SCAB would be for someone who is kind of in between in terms of computational experience and willingness, right? So there are some tools that are kind of plug and play, not plug and play, but like a GUI, right? This is not that. It's someone who's a little bit comfortable with programming, but we want to be able to take you straight from Cell Ranger all the way through everything else you'd like to do without having to do a bunch of intermediate steps and a bunch of intermediate programs. So if you read your data using SCAB's read 10x matrix and you pass just the data that comes out of Cell Ranger, which would be the counts matrix, a FASTA file of your BCR sequence data, and Cell Ranger's summary CSV, which includes things like the number of UMIs per chain and all of that information, we will automatically annotate your BCR data using AbStar. And by default, we will use the AIRR formatted annotations. So when you're done with this one function call will give you back an AND data object, which is fully populated with your gene expression data, feature barcodes, cell hashes, and your annotated BCR and TCR data. So from there, what else can we do? Well, one of the first things that we like to do is if we use cell hashes, we will then demultiplex those data. So we then have a corresponding function. And so following along again with the kind of the ScanPy API style, you have scab.tl. So .tl would be tools that we would use. And then so there's other variations of this as well. So for one of them would be, for example, PL would be plots. So there's in many cases, tools and plots that are linked to each other. So you can have a very concise API that's pretty self-explanatory when you're using it. So all we need to do now to demultiplex cell hashes is call the demultiplex function, and you pass the AND data. You can also pass it a couple of optional arguments, which are not required. And the process we use for demultiplexing is similar to a Gaussian mixture model, but we found it to be more accurate in practice. So we found that Gaussian mixture models can start to fail when you have individual cell hashes that are low frequency, below about 5% of the number of cells in your sample. So if you're working with anywhere between 10 and 12 cell hashes and the populations aren't almost exactly evenly distributed, you're going to find some of your populations that are less than 5%, and you're going to start seeing potentially issues with a Gaussian mixture model. So what we do instead is we calculate, so this is example cell hash data. So this is log2 transformed UMI counts of a given cell hash. And we can see there's two very clear populations here, a negative and a positive. What we do is we essentially just construct a continuous distribution of this UMI counts, the log transformed UMI counts, find the quote unquote global, it's actually a local minima, but it's actually the lowest local minima of this entire distribution here. And we found that as a general rule, we can find that cutoff, and it seems to be quite accurate, even going down to cells or cell hashes that are present in far under 1% of your sample. So an example here is what people have called the cell hash ridge plot. And what we can see is all of those cells that were classified as cell hash 1. We can see that if we look at the counts of cell hash 1, they're between 9 and 10 log 2 UMIs, whereas we have very clear distribution between those and all of the other cell hashes that were present in the sample. And a UMI or a UMAP clustering of the same data, which again shows variable numbers of cells per sample, shows that we get a very clear distinction of even very rare samples using this method. So the next tool we've incorporated into SCAB is the ability to classify B cells by antigen specificity. And that uses the classify specificity function. Again, all we need to pass to that function is our AND data object plus a list of antigen barcode names. And those names would, of course, be in the AND data object. So the way we classify antigen specificity, we use two data sets to do that. One is cell-containing droplets, which are identified as cell-containing by Cell Ranger. And we also then use empty droplets. So these are droplets that don't contain a cell, and that lets us establish a background for our antigen barcode. It allows us to set a background threshold for our antigen barcodes. And we use as a cutoff by default is the 99.7th percentile of our empty droplet background, which is 3 standard deviations above the background. And we found that cutoff is extremely accurate. By expressing randomly selected B cells that were classified as antigen specificity using that threshold, we're seeing 90 to 95% of those B cells actually are antigen specific when we express them recombinantly and test them. And one of the other things that we think is actually pretty interesting, because one of the things that we like to look at is breadth in terms of antigen binding. So one of the issues we've seen is that if you have multiple antigens that are potentially competitive, so going back to the flu example, if you have 7 different HA, HA1 through HA7, if you have a B cell that can bind all 7 of those, you're essentially limited by the number of BCR molecules on the cell surface. So if you are able to bind all of those, the relative numbers of each one of those antigen barcodes is reduced because they're competing with each other. So what we have found is that in some cases, the most interesting cells, the broadest things you'd be most interested in, are the ones that tend to get missed by falling below that cutoff. So what we can do to prevent that is we've used this concept of specificity groups. So what we can do is just group antigens by specificity. So for example, if you had your 7 HAs, you could just call that your influenza group. And any cell that's positive for that combined group as a whole, if you sum the counts of all of those and it's positive for that as an entity, you'd be considered antigen positive as well. So that allows us to use very large panels, which presumably compete with each other, but we don't miss any antigen positive cells because of the dilution of antigen barcodes by competitive binding. And we've seen, this is just some example data from a small SARS-CoV-2 experiment we did. And so we do see extremely reproducible antigen barcode data. So in this case, we took 4, in fact, this was an old enough experiment that we were calling them by the country names, not by the Greek letter names at this point. So we used the wild type SARS-2 spike labeled with 2 different UMIs, and 2 different fluorophores. So, sorry, 2 different antigen barcodes and 2 different fluorophores. So PE and APC. And as you can see, for wild type, we had really good correspondence, and of course for all the variants as well, we had really good correspondence between individually barcoded antigens. And so, as I mentioned before, we did some expression studies on just some randomly selected antibodies. This is a panel of just, I think, 30 or so SARS-2 positive B cells. And for those, again, we see about 90 to 95% of the B cells we pulled out as antigen specific actually were able to bind antigen when expressed recombinantly. So last kind of big function that I'm going to talk about as part of SCAB is we've also integrated Clonify, which is our tool for assigning BCR clonal lineages. So this has already been published. This is not a new tool, but integrating into a single cell data like this, I think is nice, because it's one of the first things we'd like to do when we're looking at this data. So briefly, we just use CDR3 edit distances and also shared somatic mutations, which allows us to assign lineages accurately, even when these lineages are fairly somatically mutated, as in HIV and some other chronic pathogens. And so again, as with most of these tools, the assignments are added to the adata.obs data frame. So now this data is still all linked back together into the same package, and we can work with that package very easily. So this, again, I mentioned earlier that blatant stealing of other people's good ideas. So this visualization format is essentially taken directly from nClone, which is 10x Genomics. It is an internal tool that they've been using for a while and was just published a couple of days ago and is publicly available. And so what it shows here very briefly is gives you some summary information about a single lineage. So it gives you information about the germline gene, somatic mutation for both the heavy and light chain. By default, Clonify will assign lineages names using a mnemonic. So it's just a series of 6 words, which is, I think, easier for us to mentally process than like an alphanumeric string. And what we do here is along each one of these lines is all of the sequences grouped by amino acid identity, right? So in this case, there's only one sequence in each group, but if we had several sequences in this group that were amino acid identical across the heavy and light chain, those would be combined together and shown on a single line. So as an idea of what's being shown here, this is the number of sequence, this is the count. So in this case, there's 8 total and 1 in each group. All of them are IgM. This first bin for HA is the call for antigen specificity. So yes or no. So in this case, one of them was not considered antigen positive, while 7 of the remaining were. The second column here is the log2 normalized UMI counts for each of these. So the first number is the mean for all of the sequences on this row, and the second is the standard deviation. Of course, in this case, all of that is 0 because these contain only a single sequence. You can clearly see that many of these sequences are in the 8 to 9 log2 range, which corresponds to something like 250 to 500 UMIs per cell, which is very, very robust in terms of the number of cells that are the number of UMIs that are bound, and also extremely high above background, which is in the 1 to 2 log2 range. So we're talking about a background of something like 2 to 4 UMIs and a positive signal of something like 500. So we're seeing a very good distinguishment between antigen positive and antigen negative B cells in this case. So just as a kind of a summary, so this would be all you would need to do if I wanted to go from some raw data straight out of Cell Ranger, and I wanted to find all of the B cells, all of the lineages and B cells that were able to bind across multiple HAs. This is all the code you would need to do, starting with both your the AND data, the filtered matrix that Cell Ranger outputs, which includes the cell-containing droplets, as well as the raw matrix. You demultiplex, classify the specificity, assign lineages, and then just pick out these cells which are positive for H1, H2, and H7. And now you have this nice, concise data frame of your broad antibodies, which again is linking together gene expression, demultiplexing data, specificity assignments, as well as other things that you may have as well, like heavy and light chain sequences and any other annotations you have there too. So with that, I want to talk about actual application of this tool to real data from some samples from Lassa Fever survivors. So I want to give a little bit of background about Lassa Fever in general, and then talk about the work we've been doing for the last couple of years. So Lassa Fever is endemic in West Africa. So we can see shaded here as the Lassa Fever risk zone. So at the center of that risk zone here is Sierra Leone, is the blue country there. And Kenema Government Hospital, KGH, is marked there with a star. So we have a close collaboration with Kenema Government Hospital, which is treats the largest number of Lassa Fever cases in West Africa per year. So Lassa Fever is predominantly spread from rodent to human, mostly fecal oral. So it's typically either eating a improperly cooked rodent or rodent infestation of food supplies and then subsequent eating of the food. So there's hundreds of thousands of cases in West Africa per year, but overwhelmingly they're mild or asymptomatic. Something like 90%, it's estimated, are no worse than any of the other febrile diseases that are common in the area: flu, dengue, malaria, those types of things. And are thus essentially clinically indistinguishable unless you can do virus-specific testing. So in some cases, though, it's estimated at about 10% of cases, the disease gets much more severe. And among people who are sick enough with Lassa Fever to be admitted to a hospital, the case fatality rate can be quite high, 20 to 70%, depending on a number of factors, including location and resources that are accessible at the hospital. So there are also serious long-term sequelae resulting from Lassa Fever infection. So although there's a huge number of asymptomatic or mild cases, the severe disease is extremely severe and causes substantial mortality, as well as these long-term sequelae, even in the people that are able to survive. And so we work with patients in Sierra Leone, and this is in the Lassa lab at Kenema Government Hospital, is a sign which uses Creole, which is one of the local dialects, to remind people, no eat denerata, you get Lassa. So don't eat these rats will give you Lassa. But you don't see Lassa in these rats. So if you see rats that are that shape, you can eat them. If you see rats that look like this, please do not. So the virus itself is an ambisense RNA virus with 2 genome segments. There's only 4 genes, the glycoprotein, nucleoprotein, a zinc binding protein, and a polymerase. And there are, well, until recently, there have been 4 lineages of Lassa, but there are 2 new lineages that have been discovered in the past 5 to 10 years. And the Lassa lineages are essentially geographically localized. So in our case, we're working primarily in Sierra Leone, at which lineage 4 is the dominant lineage. And there are other lineages scattered throughout West Africa. And so if we look at the Lassa glycoprotein itself, which is the protein we are most interested in because that's the target for antibodies, it's the only protein on the surface of a Lassa virion. So it's a class 1 fusion protein. If you're familiar with antibodies against glycoproteins, a lot of this will sound pretty familiar. There's 3 subunits, a stable signal peptide, which interestingly, in the case of Lassa, the signal peptide actually remains incorporated with the glycoprotein, even following cleavage and uses the signal peptide. It also includes GP1, which is the host receptor binding protein portion of the protein, and the GP2, which is membrane fusion. So it forms a fairly standard looking stalk and a stem with the bulbous part of the protein, which is the receptor binding portion. So this is a fairly standard looking glycoprotein if you're used to using antiviral antibodies. So one thing to note though is that there are a huge number of arenaviruses, both old world and new world, and the old world and new world arenaviruses use different receptors. So it's not immediately apparent that receptor binding site antibodies will be broad enough to recognize both old world and new world antibodies because there's pretty significant variability at the receptor binding domain. Although, if you look at the homology between old world and new world arenaviruses in GP2, it's much, much more similar than you would see in GP1. So the assumption is the broadest antibodies would recognize epitopes present on GP2 versus GP1. So if we talk about the breadth of Lassa antibodies in general, as it turns out, most antibodies, or at least a fairly large number of antibodies, are able to recognize multiple Lassa lineages. So it doesn't appear, so it seems to be something like we see with dengue, where cross-reactivity across Lassa lineages and in dengue serotypes is common. So this study was done using samples from patients in either Sierra Leone or Nigeria. And in both cases, they saw plasma cross-reactivity across lineages that are not prevalent in the geographic region from which the patient was derived. So for example, in Sierra Leone, there was cross-reactivity across lineage 1, 2, and 4, and same thing, 1, 3, and 4. And in Nigeria, the same was seen, despite the fact that lineage 4 is not circulating in Nigeria, and lineages 1 and 3 are not circulating in Sierra Leone. So to give you a little bit of background on what's known about Lassa antibodies, the answer is not a whole lot, but here's a couple of papers that have been published in the fairly recent few years about Lassa antibodies. So in this study, mice were immunized with what's called soluble GPCE, which is essentially just the GPC protein with the transmembrane domain and the cytoplasmic domain removed, and a fold-on appended to the end. Mice were immunized, 6 hybridomas were generated, and 6 antibodies were recovered. Interestingly, all of the antibodies they found were GP2 specific. So it appears that GP2 is highly immunogenic, at least in this particular experiment. So all of the GP2 antibodies, for the most part, were fairly broad. So these are not Lassa lineages. We're showing different arenaviruses. So the ones with an OW in green are old world, and the ones in orange are new world. So it does appear that you can develop antibodies that are broad across both old world and new world arenaviruses fairly readily, as long as they target the GP2 region. Unfortunately, all of these antibodies were non-neutralizing. So the authors speculate in the paper that GP2 the GP2 region in this sGPCE construct may adopt a post-fusion conformation. So you're recognizing a form of the protein that would not necessarily be present on the native spike, or if it is present on the native spike, is not on a functional spike that is capable of performing fusion. So there was some a hope that these antibodies maybe then would potentially have some sort of effector function that could be protective despite their lack of neutralization. And unfortunately, that was not the case. The effector function, which was seen and quite significant, did not provide any protection against challenge in animal models. So another study which used a fairly similar construct, recombinant GPCE, but this did have a stabilizing mutation, and portions of the GP2, a little bit more of the GP2 was removed. So it was a slightly different construct. And the hope was that this construct would do a better job of stabilizing GP2 than the previous study. So this was done using 17 donors, mostly from Sierra Leone, but a couple from Nigeria. And they recovered 113 monoclonals. About half of the antibodies were able to recognize GP2. But again, all of the GP2 antibodies were non-neutralizing. So again, despite the hope that this construct would potentially present GP2 in a more native-like structure, it does appear that either GP2 antibodies aren't neutralizing, or that the GP2 conformation in this antigen bait was post-fusion. So there were 16 neutralizing antibodies that were found. All of them were able to recognize, were not able to recognize GP2 alone. Only 3 were able to recognize GP1 alone. The remainder needed the entire construct, which included both GP1 and GP2, which is fairly interesting. So looking at the neutralization of these antibodies, in agreement with what we saw for the serum studies, most antibodies are able to cross-react across Lassa lineages. And so that's, again, similar to the phenomenon we see in dengue, but I think in this case, even more so, because the drop in neutralization across the lineages is minimal, right? So for the most part, if you can recognize one and neutralize one, you neutralize all 4 Lassa lineages that were tested here. And if you look at binding breadth, the red antibodies are shown here in neutralization. The red antibodies are neutralizing and the black are non-neutralizing. We did see some reactivity between Lassa and a fairly closely related old world arenavirus, LCMV. But the broadest antibodies, which recognize some new world viruses as well, were all GP2 specific antibodies and of course non-neutralizing. So again, it does appear, like we saw from both the alignment data and from the serology, antibodies that can neutralize tend to be broad across Lassa, but those that target GP2 are the broadest, but tend to be non-neutralizing, primarily because we think these the antigen baits being used are suboptimal. So one interesting antibody from this study is named 37.7H, and it's actually really, really kind of cool because it binds to Lassa in a way that's somewhat unique. So it binds at the interface between 2 protomers, and about half of the epitope is on each of the protomers, which is, again, why we would need to have, in this case, both GP1 and GP2 present, because the epitope spans GP1 and GP2, and it also spans 2 protomers. So interestingly, when this structure was generated, the GP construct that was used was a monomer, and the antibody itself actually forces the monomeric GP into the trimeric form. So the antibody stabilizes the trimeric form, at least in this crystal structure. Promisingly, 3 antibodies, which are similar to 37.7H, were able to completely protect in a animal challenge with wild type Lassa. So promising in the sense that we could use these antibodies as something like a therapeutic, or we could even use them as templates for vaccine design. So we wanted to go farther. So again, so this is a picture of our the Lassa lab at Kenema Government Hospital, where all the subsequent work that I'm going to talk about now has been done. So KGH is the largest hospital in Eastern Sierra Leone, and it's the Lassa, the equivalent of a center of excellence in the U.S. For Sierra Leone. So any Lassa cases in Eastern Sierra Leone are referred to KGH for treatment and diagnosis. KGH has the only Lassa isolation ward in the country. And so of the patients that come into KGH, about 15% of adult admissions are for Lassa fever. And we also have located here a high containment lab, which is shown here. This is kind of a wide picture of the hot lab at KGH, which is where we did a lot of this work. So working with Andrew Ward and a graduate student in his lab, Haley Parrott, they've developed a stabilized version of trimeric Lassa GPC. So these are just negative stain EM images, and this is the Lassa GPC linked to a fold-on, which is similar to the constructs used in the previous published papers that I mentioned before. And you can see that just like with something like HIV, you don't actually get trimers. You essentially get 3 monomers that are tethered together by that fold-on. So you have essentially a trimer that's fallen apart. You have like a 3-leaf clover, not an actual trimer. The stabilized trimer, however, forms a nice, tight, symmetrical trimeric shape, which preliminary structural data shows that this actually matches very closely to the trimer that was recovered with 37.7H, which was forced into the trimeric conformation by the antibody binding. So what we did is we used a small sorting panel. We used the GPC fold-on construct with a dually labeled PE and APC, and with separate antigen barcodes on each. The stabilized Lassa GPC, a positive control antigen, which was typically we use influenza HA. The positive control would be for an antigen we'd expect almost everyone in the cohort to have antibodies against. And we also use what we call a blocktail, which is a cocktail of blocking reagents that allow us to recognize sticky or potentially non-specific cells that are binding to antigen, but also binding to our blocking reagents as well. Again, these are, there's no fluorophore attached, but there is an antigen barcode, so we can detect them from the sequencing data. So here's an example of what one of these sorts would look like. So you have your double positive population here, which would be Lassa GPC. And again, since all of our antigens are labeled with the same 2 colors, we can't distinguish from flow cytometry alone which particular antigens these are binding. So if we had an antigen panel that had many HAs, for example, or many different Lassa variants, they'd all just be antigen positive, not necessarily positive for each antigen. And then our positive control antigens are down here because they're labeled with PE only. And as we'd expect, there's no APC only population because we had only dual labeled antigens or PE only. So we, in this, from 7 donors, excuse me, this is in a small pilot study, which I think, fingers are crossed based on the score, but I think we're going to get an R01 funded to do more of this work. We recovered about 820 antibodies. If we look at the genetics, so a couple of things stand out. VH12, 169, and 321 and 315 are significantly increased above what we'd expect the random repertoire to look like. So we do see some skewing, particularly in VH321, in the Lassa-specific antibody population. We also see a moderately skewed CDR3 length distribution. So we have this kind of bimodal peak here. And actually, it's not quite bimodal because one of these peaks here is a really expanded lineage from a single donor. So it actually, if you limit them to just what the clonal lineages look like, we see a peak more like around 17 or 18 amino acids, which is between 3 and 4 amino acids longer than you'd expect from the total repertoire. Also, interestingly, the majority of the antibodies were unclass switched, IgM. So that's a phenomenon that's been seen before in Lassa. There does seem to be some sort of inhibition or suppression of the immune response by Lassa. So you tend to get far less a much slower development of class switch neutralizing antibodies. And in some cases, people never develop class switch neutralizing antibodies, even though they have neutralizing IgM antibodies. So there does seem to be some sort of inhibition of class switching and normal B cell maturation in Lassa infection. So if we look at the clonal expansion, so we found 80 lineages from our 800 antibodies, 80 expanded lineages. So this is 2 or more members of the same lineage. That comprises 274 of the antibodies. So the majority of the antibodies we pulled out were singletons, which is not uncommon in experiments like this. We identified expanded lineages from all of the subjects. And interestingly, the expanded lineages were predominantly unmutated. So over half of the expanded lineages had no somatic mutation, despite the fact that we saw multiple members of the same lineage. And in fact, the largest lineage we saw here with 21 members, all of them were entirely unmutated. So they were all IgM and they were all completely germline, which is surprising and unexpected for a lineage that had undergone so much expansion. So we can also now start to look at specificity using our pool of antigens. So this plot was made, again, with SCAB. So this is one of the ways we use to plot antigen specificity with 2 antigens. So on the x-axis here is the trimer. On the y-axis is the fold-on, which we are treating essentially like a monomer, because we have essentially 3 monomers tethered together, but not necessarily in a trimeric form. So we can see 3 major groups: trimer specific, binding to both, and monomer specific. The majority of antibodies, interestingly, were trimer specific, which suggests that the previous studies that have been done using monomeric versions of GPC had missed a large fraction of the antibody response. One thing to note here before I move on, though, is this kind of extra little bubble here, which we would call a trimer preferring. And I just wanted to bring that to your attention, and I'll talk about it in just a second. So evaluating some of the expressed antibodies, this is just 2 that are shown here. We expressed a handful of them and have expressed a bunch more that's in progress. But one thing to note here is that 432.2, this antibody was thought to be trimer specific based on the antigen barcode data, and 432.12 was bispecific. So again, that's down here is trimer specific, and trimer and monomer is up here. And expression and BLI shows that that's exactly the case. We had very good binding of the green antibody to the trimer, but minimal to the monomer, and we saw binding to both by the antibody that was purportedly bispecific from the antigen barcode. So again, the functional data actually matches the antigen barcode data that we're using. So I want to narrow in on one antibody, and I'm almost done here. So this antibody called S105.7 recognizes that trimer preferring epitope that I mentioned right here. So it's right in the middle of that odd bubble. So what's really interesting is that antibody structurally is essentially the same epitope as 37.7H. So in gray is an EM reconstruction of S105.7 bound to Lassa GPC, and in red is the crystal structure of 37.7H bound to GPC. So it's that actually makes a lot of sense when we think about it, because this is that antibody that recognize an epitope that spans protomers. So it can bind the monomer, but it binds the trimer better because the trimer has both of those protomers there. So if you're binding to just the monomer, you're binding only to one, you're only using one portion of the epitope for binding. So again, even in this kind of odd case, the antigen barcode data very well recapitulates what we actually see functionally as well. And the hope, so we're still working on getting a pseudovirus neutralization working reliably, but this does bind to the same epitope as 37.7H. 37.7H is a potent neutralizing antibody. And in fact, this S105.7 is significantly higher affinity than 37.7H. So we expect this antibody to be quite potent as a neutralizing antibody. So last slide. So we actually, this is the summary slide's outdated. We actually have over 800 antibodies now from 7 convalescent survivors. The majority of them are trimer specific. Our hope is that the stabilized construct does a better job of presenting pre-fusion GP2, and that a lot of the trimer specific antibodies are in fact GPT specific and are recognized in the pre-fusion form of GP2, which is not present on the monomeric form of GPC. Really quickly, some quick acknowledgements. So Jonathan Hurtado is a postdoc in my group who has done a large amount of this work. And I need to mention our collaborators at Kenema Government Hospital who have done a huge amount of work with us and have been fantastic in helping us collect samples, process samples, and build capacity at KGH to do single cell work. And I'd be happy to take any questions. I know I went a little bit longer than I'd hoped. So if you want to get started right away with Andreas, that's fine. We can do questions towards the end.
Oh, no. Yeah, thank you very much. That was great. And we have time for questions, definitely. We'll start Andreas at the top of the hour. Don't have any in chat. If people want to raise their hands or just jump in there, I've got a couple questions, but see if... I mean, going back to like your introduction to the tool and everything, I mean, do you see much? You mentioned that, like with I think for your rearrangements, you can use AirTSV format or something like that. Do you see much with the single cell and the GEX, much hope for even more community standards and sharing and stuff? How do you see that going with your experience?
Yeah, so I think there's the standards are kind of evolving a little bit out of necessity. So for single cell and just RNA-seq data in general, there are, I think, a few standards that are generally widely used. Loom is a pretty common thing. Just a .mtx format is pretty common. Like HDF5 files are pretty common. And a lot of the tools that people use can kind of take in all of those. So in terms of like the GEX kind of stuff, I think that's already there. It's in pretty good shape. Yeah. And I think the thing that we wanted to do was not necessarily like invent a new standard or kind of make a new standard, but to try to combine the standards that exist in a way that makes things functional, right? So we wanted to take the formats that are common, like an AND data type format for Python, and take AIRR data and link that together, not try to make some new standard that combined it all, right? So that way, if you have data in AIRR format and you have other data in kind of standard GEX formats, we can just put them together. We don't have to worry about trying to put that into some new format that's now the new standard for combined data, right? So that was kind of the hope, is to make it flexible and adaptable to what people in each field decide should be their standard.
Okay, we starting to get some questions, but I'm going to be selfish. So you're working with this African population. It's funny, as soon as you put down, you're doing annotations, I go, well, what's the reference genome? And then I was wondering, well, especially with the African populations, do you think you're a problem not having a good reference for those? But then again, you've got all these non-mutated, so you must have some of alleles in the reference genome that are getting replicated. So
yeah, so that's exactly right. So that's one of the things we haven't done bulk sequencing on these subjects because we've been essentially grabbing it all and using it all for antigen specific stuff. But that's one of the things that's on our to-do list, is to generate some bulk sequencing data and using either any of the repertoire or the allele inference tools that are out there to kind of build a reference database. So we're actually, as a kind of a side project, we're working on like a nanopore-based method for IG locus sequencing. So that's kind of in progress. And one of the ideas was to test that out on our, and that's a cohort of African samples. But I think anything that involves that new method would also involve doing inference from recombined data as well. So we're in process of doing that as well. Yeah.
And that 321, does that show up with some of these clusters against the trimer, or you had these?
Yeah, so I cut out some of the data because I was long already as it was, so I trimmed a little bit of that out. But in fact, the antibody that I mentioned towards the end, S105.7, is a 321 antibody.
So Kira asks if this tool that you talked about can be applied to TCRs. We're getting a feedback from somebody. I don't know. But
yes, it can. In fact, so if a second, I'll scroll back through and go back to the first thing that I showed here. So in this read function here, there's also if you just switch BCR for TCR, you can provide BCR path, BCR annotation from Cell Ranger, and the BCR format, which can be either FASTA or a pre-annotated format. So in fact, you can apply both of them. So if you sequence like PBMCs, you can supply your BCR sequences and your TCR sequences. It'll annotate them all and add them to your gene expression matrix as well. So you can include all of that. So from like more of a technical side, the BCR sequence data is in the AgBCs data frame under the column BCR, and the TCR data is under the column TCR. So you'll have then, when you finished, an AgBCR, AgBCR columns, which from there you can get that data. Yep.
Okay. So yeah, anybody want to jump in here? I've got some other questions, but don't want to be. Anybody else jumping in here?
I mean, maybe I'll jump in with a quick question. One of the things that I was wondering, when you reconstruct the clonal lineages, is there any, given that sort of the recombination statistics can sometimes give you clonal ancestors that can just by chance be relatively close. So if you're just sort of grouping things together by CDF3 sort of editors, is there any worry that you might be grouping things together that are actually different recombination events, or do you have other information to sort of keep those apart?
Yeah, so it also includes the V gene and the J gene, right? So if it has a different V or J gene, those are, I mean, technically you could manage to squeeze them in, but it's pretty heavily penalized, right? So, the reason it's not completely excluded is the possibility that you might get a different assignment because of heavy somatic mutation, right? So one might get called 330 and one might get called 330-2 or something, because you have some mutation, but it'd have to be really, really strong alignments to overcome a different V or J gene. So that's in there as well. So actually, I cut this out of the talk too, because I ran out of time, but I think I have it towards the end here. I have some slides after the acknowledgements that I cut out. Actually, maybe I didn't save it here. Let's see here. So this is actually an interesting story. So this is the single lineage that was the largest lineage that we saw. It's interesting because we think it actually is two different convergent lineages that are extremely well converging. So this lineage is entirely unmutated in both the V and the J gene. But if you look in the CDR3, 20 of the CDRs are identical, this sequence. One of them is different at 4 positions, which again, if this V gene is completely unmutated and so is the J, I think it's unlikely that we get 4 sequencing errors all in the CDR3, right? And so it does look like that may switch where the D gene is most likely to be located. And it also potentially switches where the J gene is located as well. So this might be one of those cases where, of course, this is an extreme case. So the dashed lines here, these are synonymous changes. So the CDR3 is identical except for one amino acid. So in that case, I mean, I don't know if there's any methods that would exclude these as not being clonally related because they're so similar. But yes, we do think that there is potential for convergent lineages or mistakenly identified convergent lineages as well. Yeah.
Yeah. Okay. This is super cool.
We got one bit. I'm almost avoiding to ask this question, but maybe it's my ignorance. But in this work, was the gene expression work that important? Did it inform it that much, or was it more just producing the lineages, or where did the single cell GEX really come into play? Or
yeah, so for this particular experiment, we care about the single cell gene expression because we do get this strange inhibition of B cell maturation in Lassa. So we wanted to look and see if that's actually the case. So what's interesting is, and this is only really possible because we had the gene expression data. And again, I didn't go into all of this because I was already, going pretty long. But for this particular lineage, we just talked about this lineage with it looks like it's convergent. It's 20 BCR, 20 cells, right? And that, so having single cell helps us count that these are actually different cells and not just RNA from the same cell, which is great. But if you look at the gene expression profiles of those B cells, they're phenotypically naive, which is shocking because they don't make CD27, so they're not IgM memory, but they're expanded. So something weird is happening at like the phenotype level of the B cell, where you can expand, you don't class switch, you don't somatically mutate, and you don't phenotypically transition to a memory B cell in the ways that we would understand it. So in that case, we do see some interesting data that we can pull out from the gene expression data that we'd miss if we just did like single cell antibody PCR kind of stuff.
That's a nice example, because people I think are kind of wondering about what we get there. That's great. Well, thank you again.
And yeah, it's true. In a lot of experiments, gene expression data may not be especially useful. So it's not always necessary to do. And of course, with something like 10x, you don't have to do gene expression library and sequencing. Also, you can just do VDJ and kind of these antigen barcode type stuff if that's the preference.
Cool. Well, thanks again. That was great.